And how do we construct this library? Basically, it's placed into plasmids. The plasmids then replicate in bacteria, and the bacteria are plated out on a petri dish that contains X-Gal and ampicillin. This is done by Rutgers University. And I want to go back to a step. Here we use reverse transcriptase. We actually work with mRNAs, and they actually create a double-stranded copy, a cDNA. There are blue colonies here because some plasmids may actually get through this whole process and not ever have a cDNA fragment ligated in. If that bacteria makes it to the petri dish, it will still have the LAC-Z gene intact, and it will be able to break down X-Gal. So it will turn blue. Okay. And the construction of the library, like I said, has been done by Rutgers University. Here are some differences between the cDNA and genomic libraries, so you understand that genomic libraries, one of the biggest differences is that they have introns in between the exons. A cDNA library only is working with exons. We're using the mRNA, so we're only working with these exons, and we only have the open reading frames. Okay, so we are getting those areas that are actually translating into a protein. Genomic DNA has all this extra information here. So how did Rutgers University actually get the mRNAs out of the plant? They go through several steps to actually remove the DNA and the RNA, and they actually process out the DNA, so they use DNase, which is a substance, a protein that actually removes the DNA. And then they treat with phenol, which removes all the other additional proteins that might have come from the membrane. What you're left with is RNA. And most of the RNA is going to be rRNA. Some of it's going to be tRNA, but only 2% is mRNA. And that's the 2%, that's what we're looking for. Because we want to know what genes are these plants making? What are they expressing? And so they isolate the mRNAs by using something called an oligo-DT bead. This bead has thymines attached to the ends, which then will bind to the poly A tails. They pull those out of solution, and they filter out the rRNA and the tRNA, and all they're left with in the column is mRNA. So this creation of a library is quite an elaborate process. After they do this, they're not done. First, we need to prime the oligo-DT, okay? And the oligo-DT actually also has this SFIB site here. They synthesize a cDNA with reverse transcriptase, an enzyme that we typically saw, the first time we really learned about it was when we learned about HIV and how HIV actually takes RNA and converts it into DNA. This synthesis of the library, once they create the double-stranded DNA, it's now considered cDNA. But again, you notice the difference between genomic and cDNA is that we're actually now going to have a poly A tail in our sequence, which is not normally found in the genomic DNA or the genomic sequence. This is then treated, and a second strand primer is attached. This primer, SFIB and SFIA site, are going to help to place the fragment, to clone the fragment into the vector that we're using. Without these ends, we can't clone them. And what happens when we open up the plasmid, this area here, whenever you see a restriction site that has all these ends, it means that it doesn't matter what bases typically we have there. It just means that the distance has to be the same. And the position has to be the same. So in this case, the SFIA has this sequence where it splices through the A and the C and the T and the A right here. Whereas the SFIB site looks different. But if you notice, the ends before this end region are the same. So it's GGCC. This one is still GGCC, GGCC. And again, the same in reverse. It is a palindrome. So this section is going to be similar to this section. But what can vary is the center region. So some restriction sites are like that. And this is one example. Where is this SFI-B and SFI-A site on our original vector? It's in this polylinker region where I had told you that we look at the multiple cloning site. And this polylinker region has many restriction enzyme sites, which help us to find the position of your unknown when we get the sequence back from Rutgers and from sequencing. So if we insert the DNA in this region, then we know that we should find this SFI-A site. And one way to see it later when we're looking at the bioinformatics is we generally look for EcoR1, which is GAATTC, and then later ATTA. And in here, you should have the sequence and the DNA insert should be found in this region. So we're going to end right here, and you can watch the next video to learn a little bit about plasmid preps.